Okay, we have time for a few questions. Uh, okay, so there's a question here for Dr. Gauran from Gerard Umpad. My background is in tropical medicine, and I'm wondering if the method you presented can also be extended to infectious diseases instead of studying genetic sequences that renders people susceptible to infectious diseases. One can scan and analyze protein domains instead. I am also wondering if the 3D structure of these protein domains may affect the analysis, that is mirror images of a target protein domain present in a single CD factor. Hi.
Thank you, Paul, for the question. So I'm not 100% sure about my answer regarding the protein structure. So I will just answer the first part of the question regarding the applicability of the method to infectious diseases. I think the short answer is yes, but it's a conditional yes, because we know that for infectious diseases, we have to accommodate a lot of confounder effects. So I'm guessing, which is part of the future directions of the work we've done, is to incorporate covariates and other available information. In the Bayesian perspective, we can accommodate them in the prior information, but in empirical base, we can consider them as covariates in some sense. So I think that would be very much a good extension. So siguro po a quick perspective regarding what I know so far about infectious diseases, which is COVID. So the project that we're working with under the Philippine Genome Center right now is to sequence viral RNA viral samples of people who are positive with COVID. So the idea is to perform a metagenomics analysis for that. But so far, up to the level of protein domains, I don't know what the structure of the data set will look like yet, because the project has just started under the OSTPCHRD, but it hasn't like reached the analysis stage yet. So we're still in the process of sequencing, and we don't know the available data to us so far. But one of the questions which I think is related to the question being asked is there local mutation happening in the Philippines? So are the mutations spreading, are the positive cases in the Philippines, the samples, do they contain specific mutations which is geographically constrained in a sense? So I think yun po yung perspective ko so far regarding the problem.
Okay, so any other question from the participants or from the panel? Yes, from Sulaiman.
I have asked a question to Grace, just to clarify two questions. When those treatments, they just include different drugs, there is nothing additional food controlled, or are they same? They have the same food also. The second question is, how does the vaccination of tuberculosis play a role? What is the efficacy of that if you compare it with these treatments? I know that may not be a major topic, but if you can say a few words on that, I'll be very happy.
Yeah, hi, thanks, Sulaiman, for your question. I think the first question was with regards to how so in treatment of TB, our standard of care, the control group, is typically for the drug susceptible for drug susceptible TB. The control group is the standard of care. So we do have standard of cares for drug susceptible TB. we do have standard of care for that, which is the 6 months of HRZE, isoniazid, rifampicin, pyrazinamide, and ethambutol. And then for TB prevention, we also have a standard of care, which is the 9 months of INH for adults. And the question about control is a really, really very important question because of the problem of biocreep, right? We're looking at more and more new experimental regimens and comparing it with this old standard of care. And once you have been able to show non-inferiority of a new regimen, then which one would you be using as your control group once you have shown that? So one is to be really careful about choosing a control group in the next experiment. And the beauty of the risk-benefit approach is that you can be comparing any two regimens and not worry about which one, is the standard of care or not. The problem with non-inferiority trials is that, you have to have the control group you might have a problem of biocreep with using the same control group. A DOOR approach or a risk-benefit approach using a superiority framework actually is getting rid of that problem. I don't know if that's kind of getting at your question, but there are the field of MDR-TB, like the multi-drug resistant TB, the control group, the so-called control group, is a big question mark. We are having problems trying to run randomized trials with treating new regimens for MDR-TB precisely because of the problem of that the standard of care group is now becoming more, it's actually very much in flux because because of the dire need for a treatment for MDR-TB, actually experimental regimens are actually being used in the field now, but with things like humanitarian use, because, for example, new novel regimens like bedaquiline and delaminid are now being put out in the market, put out there in the field without a phase 3 trial, confirmatory trial of a shortened regimen. So the question of control group for certain types of TB disease, like MDR-TB disease, is very much a problem. And that's a big area of also clinical trial methodological research that we're trying to get around with. The second question, I'm forgetting what it was again.
Like vaccination?
Vaccination, yes. Another good question.
How effective?
Yeah, vaccine research is very much, we don't have there's no real effective vaccine actually against prevention of TB. We have this vaccine called BCG, which is now being discussed in the field of COVID. There's actually a clinical trial that's looking at BCG vaccination, which is supposedly a TB vaccine, whether it's actually working against prevention of COVID. But BCG vaccine actually is only effective in certain kinds of TB, usually the extrapulmonary type of TB, which is very common in children, but it's not very effective in adults. So vaccine research is very, very critical. And it's actually the question of whether to think about looking at vaccine as an arm and a TB preventive therapy as another arm is also one of the question marks, because those are both preventive regimens in some way. So yeah, that's a really, really good question. I'm not sure if I have an answer to that, but the one thing for sure is that there are TB prevention therapies like INH that are working, It's just that they take a long time, 9 months normally, 6 months. And now we have a 1 month regimen that's working, but there is no TB vaccine right now. And TB vaccine is supposed to be the sort of the panacea to getting rid of, the TB epidemic that's happening alongside with COVID.
Thank you.
Okay, thank you very much, Grace. We have here a comment from Edsel to Tony and Peter, very creative use of MC theory in the context of anchor theory. Also from Edsel, thanks Grace, nice applications. And there is a question here from Giselle Concepcion, our PAASE president. Iris, do you consider proteins only with high resolution structures from protein crystallography data from PDB? How about membrane bound proteins? Also for Grace, have you considered working with Philippine DOH TB teams in RITM and with TB epidemiologists in UP Manila and other TB centers? Have you accessed their quality of data they have? So we can ask Iris first to answer that, and then Grace after.
Thank you for the question. So the biology aspect of the paper was really maneuvered by our colleagues. So the one that I can answer with certainty on my end is that our source of is the domain mapping of disease mutations that was a mapping from an existing database to each disease mutation by gene or protein or domain location. So this in this mapping, the proteins were aligned to a database of conserved protein domains using an alignment tool. So that's as far as I know po. Pero in terms po of membrane proteins and other specific aspects of proteins, hindi ko po talaga i-claim na I'm an expert in that matter. So yun po.
Okay, Grace, on TB dots.
Yeah. I have not been in contact with folks, researchers in the Philippines. Would be very happy to have discussions. I'm very much interested in any potential for collaboration. So anyone who wants, feel free to send an email. In terms of epidemiology or clinical trials work, that would be very much of interest.
Okay. I have a quick question for Tony. Are you still awake, Tony? Other than marketing, any application you can envision for option framing?
Well, actually, it's especially with if you blend in Markov chain, wherein the options become the states, you can use it for promotion strategies, wherein you have this joint occurrences of different options. Then you can see what option gives rise to another option wherein it will trigger more purchase. So it's still along the line of marketing. The option framing, I think, is, I don't think you can cure cancer with it, but but it will give, it's more on marketing. I mean, first impression is everything. It's about packaging. It's about packaging.
But you could use it in presenting options to leaders, political leaders. They might decide to do something because of the way you put the options to them.
Okay.
I think in Dr. Mariano, it's been very interesting because you have this, um, I don't know, causality in your models, and I don't know if I can claim so much of causality with this Markov chains because it's just a reflection of how they behave. And in fact, the Kahneman theory, anchor theory, although he won Nobel Prize for that, is also, it's hard. The quantification of this risk could still be in question, but somehow the Markov chain transition probabilities could be made use. And the use of this option framing to verify price sensitivity results can also be used, the option framing. You can use that, I think so. I think you just gave me an idea, Ernil. Maybe I'll use that for checking the price sensitivity results.
You have to pay me a royalty for that.
Oh, sure. I will. I'll give you.
All right. Okay, so any other questions, comments from the participants, from the panelists? If not, great. Yeah.
I have a short question for Iris.
Iris.
Now, yeah. Now, do you have issues about dependent data that you're getting from this? Because you have these proteins, but you could have dependencies on those observations that you're going to use. So how did you deal with it? Or did you assume independence or positive quadrant dependence?
So that's one of the problems that we dealt with at the beginning. So part of the study from Tony Kai was dependent structure. So that was part of the literature that we looked into. But for our simulation purposes, for the initial study, we did not assume dependent structure. But for the other one, we did. So we handled that caveat of the original paper. Hence, kaya po, there's a sense of nanganak yung paper because of the possible caveats that were addressed from the initial.
Okay. Yeah.
Okay. Can we request Grace to turn on your video, and then we will ask for a souvenir photo from Pao. Pao, can you take our picture? just like in a face-to-face conference for documentation purposes.
That's a good one.
Pao, can you take our picture?
Yes, sir.
Okay.
You have to count.
Okay.
I'll join, sir.
Okay, go ahead.
1, 2.
One more.
Wait, sir. I'll just save this. Yeah. And then isa pa, sir. Okay na, sir.
Okay, so again, thank you very much, Dr. Mariano, Dr. Peña, Dr. Gauran, Dr. Marañon, and Peter, Dr. Montepiedra for a very interesting session with you this morning. Thank you for agreeing to join us at this unholy hour, especially in Sweden, Tony. Thank you very much.
Thank you very much, sir. Yeah.
Good to see you all.
Nice meeting everyone. Yeah. Grace, nice seeing you again.
Good to see you. Good to see you guys.
For the rest of the participants, PAASE, APAMS is going for the next 3 weeks. So we have 3 more weeks.
3 more weeks, yeah.
APAMS. So please register ahead of time. The talks, the sessions are posted in paase.org website. So again, thank you very much. Nice seeing you all virtually at least. Sir, thank you very much.
Okay, Ernil.
Thank you.
Thanks too.
Grace, hello.
Kamusta, Grace?
Musta ka na, Grace? Nakamute ka pa.
O, pwede na magchikahan.
Hello! Nako! Oh my gosh!
Pwede ka na makipag-chat.
Ang tagal na natin.
Kailan ka pa nandiyan sa Sweden? Matagal ka na?
Naku, bago pa nag-COVID.
Nagsasalita ng Swedish.